All right. So thank you everyone for joining. We're pretty excited to tell you about our upcoming opportunity. I'll just fly through some super quick introductions and then we will talk through what the program is all about. So my name is Heather Manley Lillibridge. I'm the Executive Director of the Cross-Border Threat Screening and Supply Chain Defense Center of Excellence here at Texas A&M. That's a mouthful. CBTS for short. We are a DHS funded university-based center of excellence. So the project funds are coming to us from Homeland Security. My colleague, Chris Camardo, is on the phone. He is our program manager and director of finance. And then I'll let the other folks introduce themselves a little bit later in the presentation. But we have three faculty that have really helped bring this Summer Research Institute into fruition. Dr. Brick, Dr. Mondati, and then Dr. Krowski, who hopefully will be joining us here shortly to talk about the different projects. Can everyone see my slides? Okay, great. So this is a Summer Research Institute in Biothreat Defense. I'll tell you a little bit about more about what we are and what DHS is if you're not super familiar with them. So the centers of excellence, there are actually 7 current centers of excellence across the country. There is a congressional mandate for up to 10, so there might be some other folks joining us. And they're all focused on research and education in mission relevant items to the Department of Homeland Security, but we each have different focus areas. And then whoop, for Texas A&M, we actually have 2. No other university has 2 centers of excellence. So we have one that's active, which is CBTS, and then we have one in infectious animal diseases called IAD that has passed its 10-year period of performance. So it's called emeritus, but it's still active and doing research and education projects. And if you want to learn more, there's a link to see what the other centers of excellence are doing. So a little bit of background on the Department of Homeland Security. It was established way back in 2002. And it basically took a lot of disparate organizations and combined them under one federal organization to help coordinate emergency response and homeland security functions. We, the Center of Excellence, actually fall under something called the Science and Technology Directorate, or S&T for short. That's the research arm of Homeland Security. So groups like FEMA or Border Patrol, they don't necessarily always do their own research. A lot of that research for new projects on the or new products on the horizon is done by S&T. And under S&T, this is more information than you need, is an organization called Office of University Programs. But it's very important to us because those are the folks that pay our paychecks. And on the slide, you can see a bunch of their different logos. These are a number of the organizations. They're called components that we either have or will be supporting. So such as Border Patrol, the Transportation Security Administration, FEMA, and a few others. So our purpose, as I mentioned before, we all have our own little niche focus areas. We're really focused on biological threats and building resilient supply chains. And we're focused primarily on the southern border, but we also do work with the Canadian border as well. A lot of us in Texas, we kind of forget about our neighbors to the north. And what we are trying to respond to biological threats, and I'll tell you a little bit about what I mean by that. So these primarily for our center are human health, agricultural health, So agricultural health, that encompasses both livestock animals as well as crops and other plants. So threats that come across our border every day include cattle fever ticks, which is important in one of our research projects, and you'll hear a little bit more about that. And then there are also a number of invasive species and agricultural pests that can come through in containers or in trucks as they come across our border. So it's very important to find these before we have a huge outbreak of disease, which could have a disastrous impact of our economy. You may have heard of things like foot and mouth disease. Thank goodness we don't have it here yet, but something like that would completely decimate our agriculture. We also do a little bit of work in other supply chains related to human health. We've done some work in COVID pandemic, the shortages of masks and other personal protective equipment. And then we are currently investigating supply chains related to pharmaceutical products. Here's our website if you want to do any more deep diving into CBTS and what some of our projects that are currently being conducted or have recently ended, cbts.tamu.edu. So now the important stuff. Let's get into the program a little bit more. It's a 10-week program, and it's going to be actually hands-on in the laboratory, one of two locations, either here in College Station or down at AgriLife Research and Extension down in Weslaco. And then we are going to have travel between the two spots. And don't worry, you're not going to have a car, or if you were in college, I didn't have a car, or in graduate school had an old clunker car. You don't have to worry about it making it that far. We're going to handle travel for you all. We do have a student stipend. We don't want you to have to work another job to pay the bills while you're doing the hands-on research. We don't have final budget approval right now, but it's going to be in the realm of $5,000 to $6,000 for the 10-week program. And that's, we base that off of the NSF, what they pay for their undergraduate programs. We're pretty excited. We're going to have a number of guest lectures. So folks from DHS are going to come and talk about their career and their career path, how they got to where they are. Sometimes you can get a job with Homeland Security directly after undergrad. You don't necessarily need to have a master's or a PhD. One of them is going to actually talk about, he used to work for the USDA before he began working for Customs and Border Protection. So I think it'll be pretty interesting. We are also going to plan a field trip. The one advisor that I mentioned that's going to talk about his career path, he's going to arrange for us to tour the Far Bridge, which is way down south, and it's the largest port of entry for agricultural produce. It's quite something to see. I've been down there and toured, and the agents down there, the agricultural specialists, are very enthusiastic about what they do. They do tours quite often, which does take away from their day jobs, but they let us look at bugs and different pests through microscopes, and talked about what the priority species that they're looking for that could really have devastating impacts upon us. We're also going to try to help you with professional development. So whether that means going to graduate school, how do I apply to graduate school? How do I put together a competitive application? What are they looking for on my personal statement? Or if you want to get a job after you finish undergrad, say you want to enter the workforce and you want to start paying off your student debt. Well, what does that look like? If you're looking for a government job, it's a little different than applying for a job in a company. They have different criteria and knowledge, skills, and abilities that you have to meet. And we'll talk a little bit about that as well, and how you even find those jobs. We also want to help you with how do you talk about what you're doing. We're calling it an elevator pitch. So if you had to present your work very quickly, and we call it an elevator pitch because basically in the time that you would take to write up or down in an elevator, can you talk and really give a snippet of what it is that you're doing? When I was in graduate school and I would try to talk to my mom about what I was doing, she would be like, I don't understand. I don't understand what you work on. So how do you get that into layman's terms? And so that's important, not only so you can tell your mom what you're doing and why you don't have a job yet, you're still in graduate school, but also if you're talking to sponsors or other people that have an interest in your program who may not be technical experts, how do you talk in layman's terms and make your project understandable in a very short amount of time? We're also going to have, let's see, some other hopefully guest lectures come in and talk about some different aspects. So I think we have a super exciting summer planned for you. So the application, this is the super important part. The deadline is April 11th. So note that it's going to be here quicker than you might realize. And that this is for all materials. And I'll talk about what all those materials are, but make sure you get everything in, hit the submit button by midnight. Later in your life, if you're applying for grants, if you go to graduate school and become a principal investigator, if you hit the submit button a minute too late, they will not accept your application if it's not in the right format. So please, please, please pay attention to the deadlines and what exactly we're asking for. If you go to the website, everything, and I'll show you the website here in a few moments, everything is laid out. The pieces include an application link. Dr. Brick helped us so much with our Qualtrics application. I'd never experienced that before. So it's super user friendly. You go in, it takes you through all of the questions one by one. Put your name in, your GPA. You'll have to answer a few other questions, but should be super easy. The harder part that you're going to have to do ahead of time, perhaps in a Word document, and then cut and paste, is your personal statement. So in your personal statement is really where we want you to kind of sparkle and shine. Talk about what it is that makes you stand out from the other candidates. What are your unique skills and abilities? Have you ever done a research project before? It's not a deal breaker if you haven't, but if you have, please talk about what you did and who you did it with. Give us a little snippet so we really have an understanding of what you've done. If you have had to work just some crummy jobs, and I packed Irish Spring, I parked cars, I did call centers, I did a lot of things when I was an undergrad. And so I didn't necessarily have research experience, but that really shows you know how to show up on time, you know how to be dedicated, and you can work hard. Talk about those sorts of things. And then talk about what you want to do when you leave school. Do you want to enter the job force? Do you want to go to graduate school? Give us a feeling of what your career goals are and how you think this project, this experience might help you later in your career goals. And then the other thing that we will ask for you is to name a reference. Please, please, please give them a heads up ahead of time so they're not surprised. The system's pretty automatic and it will shoot an email to your reference. And they will have a super easy form that they will have to fill out. If they prefer to do a recommendation letter, that's fine. But the Qualtrics system, we tried to make it as easy as possible. We also have a space to upload your transcript. It does not have to be an official copy of your transcript. If you have any problems uploading it, you can also email that, and there's the CBTS email address is provided there. Please send it as a PDF so it's easy to read and to look at. And again, Can't stress it enough. Get it in by April 11th. The other two things that I should mention as far as application goes is that this opportunity is open to juniors and seniors. We'd like you to have had a few science classes under your belt before you do this. If you're too young, don't worry. With DHS approval, we're hoping to expand this program and do this again for the next 5 years. The other stipulation, and apologies for this, but it is from Homeland Security, you have to be a US citizen in order to apply for this opportunity. So without further ado, we're going to talk briefly about what the projects are. But before we do that, I would like to ask Dr. Mary Brick to introduce herself. And then after you do that, then I will turn the presentation over to you, Dr. Mandati.
Hi, everybody. My name's Mary Bright. I am an associate professor in the Department of Biochemistry and Biophysics, and I'm also the Associate Dean for Academic Affairs for the College of Agriculture and Life Sciences. I have run summer research programs for the past, 16 or 17 years. And so I look forward to working with you all and helping you with your professional development during the summer. And so, Dr. Mandati, turn over to you.
Yeah, thank you, Heather and Mary. Thank you again, all the students and Chris to organize this entire Summer Research Institute. And I'm really pleased to be part of it. So I'm Kranti Mandati, the associate professor here at Texas A&M AgriLife Research and Extension Center at Weslaco. I'm affiliated to Plant Pathology Department, which is also on campus in College Station. But my lab is here in Weslaco, so one of the host labs of the two labs that this institute is hosting this year. Heather, should I continue with the slides, or how do you want to do it? I know.
Why do you want to talk about your project, and then sure, if you want to talk about Dimitri's, or I can wing it.
We can wait for Dimitri, maybe if he shows up by the time I finish, and then if not, we could help.
Okay.
Okay. So as I mentioned, I'm in Weslaco, so my lab here works on multiple things. In this case, for the focus of this project, this institute, we're going to focus on the pests and diseases of significance to agriculture, and also our basically across the border, we're moving in a lot of produce. As Heather was mentioning, we are the biggest port of entry for bringing in a lot of fresh produce from Mexico. So we also, at the same time, also sometimes inadvertently, we bring in pathogens or pests of great significance to our ag here. So we are studying a lot of those emerging and are already existing pests and pathogens that affect agriculture. And in this case, for the Summer Institute, the focus of this hands-on training will be in multiple areas of this broad topic. And one of them, for example, is going to be about how would you identify a pest or a pathogen of importance. I give you here 3 different pests and pathogens. And as an example, again, we have in the valley here, we are the only place in Texas that grows citrus commercially. So we have a lot of pests and pathogens that affect citrus. And one of the diseases is called citrus greening. If you have heard of it, it's pretty bad in Florida. Also, it's there. It's almost there in every citrus producing state and country. And it's affecting also the yields of how much fruits we can get in a tree. And it's transmitted by some insect vectors, just like human pathogens can be transmitted by insects like malaria, for example. In this case, the citrus greening bacterium is transmitted by the so-called Asian citrus psyllid. So we have it here in South Texas, this Asian citrus psyllid, and in many counties beyond South Texas also has it. But citrus is not grown in outside South Texas or Rio Grande Valley commercially. There is residential. You might have up in College Station a tree or two in the backyards, but which is why also it's important for us to, before we move any plants, citrus, especially from here to up north, or bringing anything from any other states to Texas, we need to make sure that we don't bring in pests and pathogens. So we're going to train students in areas of identification of, for example, how to identify citrus greening disease, both in the plants, in the trees, and also understand a little bit more in the molecular level. How do you detect it? The gold standard right now for any diagnostics in human diagnostics of diseases or even plant diagnostics, what is more standard is doing a PCR. You may have heard of it already. We're going to train the students in PCR and how would you use that to detect disease and the bacterium that's in these plants. We will also train in the lab here. We do a lot of research projects where we need to actually keep some of these diseased plants and also the infected, the vectors, the psyllids in this case, we maintain them. So the students will be also familiar with some of those aspects of maintenance of these materials for research needs. And you will shadow some of the research staff that we have in the labs. So that was an example of citrus greening, but similarly, students will have opportunity to look at other pests and diseases that affect other crops, such as potatoes. There is another disease called zebra chip, which is actually the first report of zebra chip was in North America, was in Saltillo, Mexico. From there, it moved up north into Texas, and it's endemic here. It depends on the season. The severity could be high and low, but it is also spreading slowly towards north in where potato is grown. In fact, potato is grown in many up north in Pacific Northwest regions in Colorado, Idaho. The big producers are there. There is the disease there and the psyllids are there, but it's not as still severe. But we'll give you an introduction of some of these other diseases too. There is a third one, just put there some pictures there. It's called the Pierce's disease in grape wines. We do have some production of grapes in South Texas, but most of it is in the hill country. You probably know of San Antonio, and even in College Station, you have a nice, beautiful Messina Hoff winery, right? So the disease that affects grapes is called the Pierce's disease. Again, there is an insect that transmits the bacterium, and that's the grasshopper. In this case, the glassy winged sharpshooter. We don't have much work in Pierce's disease, but we will definitely, we have some of the infected plants here, so we will be able to at least be familiar with how to identify symptoms. So that's one area of topic we will train the students in, which is disease identification, pest identification, both by phenotypic, looking at the symptomology, or by looking at the PCR. If you can advance to the next slide, please. Okay, so what I just described is exactly in this slide. I went ahead. So what you're seeing here is on the left is my technical staff surveying for this disease in some of our citrus trees here, collecting the insects that transmit the disease, and then bringing it to the lab and doing the PCRs. So you will also learn all of these, how to run electrophoresis, a gel that differentiates different PCR products. In this case, DNA. We are amplifying DNA. You may have taken some biology, microbiology, or even cell, classes in undergrad, so they may have already practical experience, but we'll show you how we do in a research setting. Another project that we may have collaborations with another lab that is Dr. Dmitry Kurowski's lab is up in College Station, is this Raman Diagnostics. We have worked together with him in using new techniques such as Raman, which is a different technique he came up with, and it's already known in other applications, but for plant diseases, he's a pioneer. And so he uses this Raman to detect diseases which without really damaging or removing the leaves from the trees. It's like a scan. It's a radar. So Raman is a wavelength that they look at specifically for signatures of disease. So you will learn a little bit of that through other projects with in collaboration with Dr. Dimitri and my lab. So that's on the pest disease and diagnostics one more time. Next slide, please. Okay, so another major area of training that we can offer to the students is, in this case, how do you maintain clean plants in terms of disease-free, pest-free plants, and how do you propagate them? If you had to plant some of these in the fields in the future, you need to make sure that you don't plant already infected, sick one, especially if you are in a place where the diseases are already there and you collect fruits and materials from existing infected trees and you would like to plant, it's not going to come out clean, right? So here what we show is essentially what we do is maintain some of our clean materials in vitro, almost like in a human cell biology or any other labs. You would have to maintain everything in a perfectly sterile condition. So you will learn a lot of microbiology, sterile aseptic techniques that we use to propagate these clean plant materials. So that's another area of my lab that works on research. So students will also get hands-on experience by shadowing with some of our research staff in these projects. So with said that, so we have a lot of undergraduate students coming into our labs, both helping in different projects, but also getting trained. We have students from UTRGV and STC and other universities here regionally. So it's a very good exposure for many of the students who go from here and get careers in other graduate schools, or even taking up jobs in industries, or even universities as a research assistants and technicians. So many of them went through my lab. So I'm really happy to receive more students. And we're in the borders, South Texas. So don't be afraid to come in if you are coming from College Station. If you already know the place, it's a beautiful place. You should, we're about 45 miles to South Padre Island. In the weekends, you don't have to work. So you can go to the island if you want to take a break from the training. But it's a beautiful place to be in. It's going to be a little bit warm in summer, but don't be scared. Okay, it's a nice place in the valley. And with that, I see Dr. Dimitri Kurowski online. So great, Dimitri, you're right on time. So we can go back to your slides and maybe you will hear from him the second lab in College Station and how we can also work together with him. Thank you.
Thank you, Kranti. I sincerely apologize for being late to this meeting. Yeah, it's definitely, I think, great opportunity for students to work on the aspects related to the national security. And in my lab, we develop Raman spectroscopy. It's a very powerful analytical approach. It can be used for non-invasive and non-destructive assessment of the structure of objects that we deal with. And we have a large set of projects related to plants, in which one of them we collaborated with Kranti. Other projects are focused on confirmatory identification of tick frass. And it is very important because different tick species, they have different chemistry of the tick frass, and by the non-invasive analysis of tick feces, we can identify tick species. And all entomologists know that different tick species vector different pathogens, and consequently, if we know what tick species cattle possess, we can conclude about the danger of pathogens that can be present in this animal. And we have a first proof of principle by our lab, and this first proof of principle shows that this technology works, that we can use Raman spectroscopy to detect and identify tick species, and through this identify pathogens that this tick species vector. And if I may come to the first slide of my presentation. Yes, so the student will have an opportunity to learn what is Raman spectroscopy, and we have several handheld instruments that a student will learn how to use and how to operate. These are really state-of-art instruments. Each of them cost about 50K, but they're really robust and reliable for student use. So we probably need about 15-20 minutes of training for an undergrad to use, but I think it's really a cool thing that you guys can use this really top scientific instruments, and you will learn how to use them. And the second thing, what you will learn is how to interpret the data, how to do spectral analysis. We'll train you and teach you what every single band means, so you will know how to interpret the spectra. And finally, we will introduce you to chemometrics. Chemometrics is the set of molecular model of digital modeling, and this digital modeling allows to do predictions about the data. Specifically, using chemometrics, we can find that the spectrum indeed came from a class of spectra that belong to healthy individuals, for example, or individuals that have the pathogens. And this is done using eigenvector and MATLAB. So routinely, our undergrads use MATLAB and use chemometrics. And again, we have a nice kind of a system of training how we can pass this knowledge to guys that will join this program. So at the end, you will know how to use instrument, how to interpret the data. So by other means, you'll be a good Raman spectroscopist. And finally, you will know how to do chemometric analysis of data, which is a very important and valuable component, because chemometrics is the front door to lots of jobs in both pharmacy and academia. And if you go to the next slide, please. Here, the slide demonstrates that, as you can see on the left side, is the results of spectroscopic analysis. Here you can see spectra that were acquired by the instrument. And as you can see, that spectra from different tick feces are different, even visually different. But what we actually do is this middle part is this chemometric analysis. And you can see the prediction accuracy or true positive rate with which the spectra can be assigned to the particular class. And these results demonstrate that we can detect and identify different tick species and their varieties. And the confocal instrument that will be also available for this work is in the right part of the screen, as well as the handheld. And my last slide is a slide of true success. Throughout my academic career here, I mentored probably more than 20 students, undergrads, undergraduate, and graduate students, and all of them, most of them, probably I should say, have very bright scientific careers as well as careers in academia. Many undergrads became doctors or dentists, and of course, you will be the next picture on this slide if you join and work with us and learn how to use Raman spectroscopy, how to use chemometrics. This will not only make you stronger, this will not only expand your scientific outlook, but I'm sure will open many doors in your careers, both in Department of Homeland Security, industry, and of course, academia. Thank you.
Okay, so I'd like to see if I can figure out how to end the slideshow. So does anyone have any questions? Feel free to put your hand up or blurt them out, or go ahead and put it into the chat and we can answer some of those. Nobody? You're a shy group. Okay, who wants to apply? I don't know if I'm qualified. Okay, well, I guess if there aren't any other questions, we look forward to your applications. We do have 6 spots available for this summer, and hopefully next year we will have even more. But look forward to reading your applications. If you have any questions, if you get into the website or the Qualtrics and you're having trouble uploading your resume or your transcripts, let us know. You can send an email to the email address. It's in the chat, cbts@ag.tamu.edu. If you have any questions at all, please feel free to reach out. If you have a question that's specific for one of our faculty members, we can relay the information, or if you have questions about CBTS or the program, please feel free to reach out to us. Well, thank you all. Oh, sorry, go ahead, Kranti.
So I just wanted to, on behalf of the students, maybe I can ask a question.
Yes. Thank
So
you.
how is the application, I guess, process after the application? They will hear from us, you and Mary, Chris, at some time. What's the time frame before they know when? You
So-
mentioned six slots. If there is more applicants, so when will they hear so they know?
Oh, okay. So the application will close on April 11th, and then we will do the evaluations, make sure that the faculty members complete their part of the application, filling out the reference forms. So we are going to review those April, and then we will make decisions in May. And we will let you know if we need any additional information, but you will hear from us either way.
And
And I'm sorry, I thought there were 6. There are 5 spots. So I misspoke.
sorry.
Oh, sorry, 5, not 6 student spots.
Oh,
I misspoke.
yeah, sounds good to me.
Okay, anything else? That was a great question. Thank you.
Sure.
All right, everyone. Well, thank you so much for your interest and your time today, and we look forward to hearing from you. Thank you, and good luck on finals.
Thank you all.
Thank you.
Thank you.
Thank you.